And Salem is assistant professor of physics at the University of Maryland for about 2 years, correct? And he uses biophysical approaches to study morphogenesis. And I believe he's going to talk about bird beaks today.
Right, so it's a great pleasure. Yeah.
Yeah, okay.
Everything's
all set.
Yeah. So can you hear me on Zoom? In here as well? All right, so it's a great pleasure to be talking about this project, which started in 2018 at the first version, at least for me, started in the first version of this workshop. And this is work I was doing with Maha and Gary, who just made it. And we were collaborating with Arhat Abzanov and Helen James on Hawaiian honey creepers. Right, so just a little bit of an overview about what I do. So like many of you, I'm really inspired. My greatest life inspiration is morphogenesis, or the process of development, how one cell turns into you and me. Or not one cell, two cells to turn into you. That's not good coding. I said that the lens I take it is through the dynamics and control of shape. So that's sort of one area is morphometrics, and really this started for me through this through working with Maha through these workshops. So morphometrics, this is very beautiful for me because I get to work with people who learn about these animals in a way that I wouldn't have learned. So it's an excuse to know about Darwin's finches, Hawaiian honey creepers, antlers, eggs and egg patterns, insects, leaves, and the fly gut. So very exciting collaborations there. And so this part is about description of shape, and then we think about how to understand shape changes and how they happen through morphogenesis. And that's from the cell scale, whether it's E. coli, to leaves, and what I'll talk about today, bird beaks. And we've also been trying to take the principles that we learn here and see if we can get them to artificial systems. So with Maha also, we've been thinking about morphogenesis in solid state batteries. So thinking about how to regulate electrochemical interfaces in analogy to how living systems regulate their interfaces. And recently I started, I got this seed fund to try to develop a digital twin that's root aware to try to model morphogenesis of plants and increase yield. So if you have any ideas on how I can do that, let me know. So I'll be very happy to talk about any of these areas. So today I'm gonna just mention the bird beak, and in terms of morphometry and morphogenesis, and focusing on Hawaiian honey creepers and Darwin finches. And these are classic examples of adaptive radiation. So that means single species. In the case of Darwin's finches, it came from either mainland South America or Caribbean to the Galapagos. And it diversified into many different species, which are ecologically distinct with many different diets and behaviors. So crushing seeds, cactus, probing cactus flowers, woodpecker, and even drinking blood. So this is a vampire finch. And over the course of a million, it ranges from 1 to 3 million years. It diversified to 15 or 18 species. Okay? And even a more dramatic example, unfortunately, Helen James couldn't make it, but she would have talked about some of this. So this is work that we did with her. It's even more dramatic than Darwin's finches and even more pretty. And the dimension that they have compared to Darwin's finches, that's even more exaggerated, is the curvature of the beak. This one, it's called EEV. It had coevolution with flowers on the in Maui, I think, and the curvature of the flower matches the curvature of the beak, and the bird can drink flowers and watch for predators at the same time. And again, we have this shows like the diversity of shapes that you get in this group and the relatives compared to their relatives. All right, so look, thinking about this adaptive radiations, the main question that comes, or one of the questions is, when do you get this evolvability? Why some species multiply and why others don't? So you can see this more clearly in Darwin's finches if you compare it to other birds that arrived in the island. The biggest number after Darwin's finches is the mockingbirds, which only diversified into 4 species, not as morphologically distinct or ecologically distinct. And other birds just stayed as 1 species. So the question is there something intrinsic to Darwin's finches that make them more evolvable versus other species, or is it extrinsic? So this shows that the fact that this is on the same island, same range, there's some intrinsic factors. And the spoiler alert, I won't answer that, but one of the some hypotheses is are they better at island hopping? So Darwin's finches have people observed them over 40 years. That's the work of Peter and Rosemary Grant. Did very beautiful work on this. Absorbed their evolution or microevolution over the course of 40 years. They saw birds and new populations being established on an island. They saw a hybridization between different species producing viable offspring and basically a new population. They saw populations coming, going extinct, and competing. So there's a lot of island hopping that sort of is something that increases the rate of evolution. But more interesting for us is how they develop. So whether they have a modular developmental program that can make them evolve faster, or whether development is integrated, so the skull could be integrated with the beak in terms of how they grow, and that accelerates the rate of a given trait, how fast it can evolve. So we're gonna our contribution is giving a way to describe the shapes of the beak and how they develop. Are there any questions so far? Okay, so Anush talked a lot about this, so I'll just give a quick sort of distinctions that I need for the talk. So quantifying shape is really hard because I think there are 2 big reasons. Is that one, shape is high dimensional or infinite dimensional. So I can give you this shape as 10 million or 10 billion vertices, and I say, here's one shape, here's the second. Like, how different are they? Or compare them. So the challenge is this: we can't, to say something about it, we need to reduce it. So that's a data science challenge. And the second challenge is that there's many ways to do that. So you're going to have to make a choice, and you want your choice to not be arbitrary or not introduce things into your conclusions. And what Anuj was talking about, the main principle for this is invariance. So if you respect invariance, then for example, you're not gonna have your conclusions depend on how you parameterize your shape. Okay, so there's several ways. Anush talked about this. You could do shape features. So this classically like measuring length, width, and depth, and or topological data analysis, or things like that. So this is shape features, but like Anush said, this doesn't give you the shape. You can't, from just the features, recover the shape. Another approach is to compare different shapes. So that's comparative. And the methods that we're gonna focus on are parametric. So I call it parametric morphometrics, which means you're gonna fit the shape that you have to a given functional form. So what's been done before in Darwin's finches is that you have, they just fit parabolas to these beaks. Sorry, I need to drink water. I'll just be there. Yeah. Excuse me, sorry about that. Yeah.
I just about the premise of the evolvability versus the number of species.
Yeah,
You could imagine potentially that the mocking birds are just,
it went extinct.
sorry,
That some of them went extinct,
or perhaps that they're able to crossbreed. Like maybe they're so good at island hopping with the gene pools and getting separated
so
such that it's more.
that's-
that doesn't necessarily imply that they're sort of ill-advantaged or ill-suited to their environment.
yeah.
Like,
So they could be suited for the niche that they're occupying. So they don't need to, or like you were saying, is that there could be enough mixing between the gene pools that they're not forming into different species.
Right, they're just not becoming isolated.
Yeah. So that's why I think the island hopping thing was through the method of becoming like going allopatric, being isolated, then sympatric. So it's like you separate and then you come back, and that leads to competitive exclusion and more processes that lead to speciation. But yeah, it's not known. The other thing is, like on average, like they could have went extinct, but that's another possibility.
Mhm.
Yeah. Does that answer the question?
Yeah, like,
All right. So the other method, which again is more important for us here in development, is to describe shape as a generative model. So what's been done here is in shells. So you can describe the shape of a shell by fitting a model of morphogenesis to it. So this is you have a coiling center line and a radius, and the parameters of how much this radius is growing gives you get the shape and the development at the same time. And so we're gonna talk about something for the beaks, understand the development of the beak in this sense, and relate it to a more parametric method of understanding their shape. All right, so that's Gary. I didn't know he was gonna be here, so he's here with us. So we're starting with a scan of the skull like this, and then the only manual process decision we have to make is figure out where to cut the beak. So this is for the part where we only want to study the beak by itself. And then we align it so that the x-axis is in this direction, in the long direction. So we fit an ellipsoid and align it like this and define this as the x-axis. And then we do some smoothing and cut it and get a smooth surface as that. So this I could do this cut when we were studying just Darwin's finches, but when we look at Hawaiian honey creepers, I can't really just cut it with a horizontal plane. I needed to have something that's adapted to the center line. It's really curving a lot, so we can't just choose a Cartesian coordinate and cut it like that. So what we did is we first take the beak and then cut it with vertical planes, and then from those planes, calculate a center line. That's the center of each cross-section. But we want, that's an arbitrary choice to cut it in horizontal planes or vertical planes. So we want those planes to be normal to the center line. So we calculate new sections that are normal to the center line, but the curve is not at their center. So we keep iterating, use the cross sections to calculate the center line and the center line to calculate the cross sections until we get normal. So these are normal cross sections, and this is their center. And so
that's the medial axis.
Yeah.
By definition.
Yeah. And so all of this stuff we could do automatically and extract these cross sections. And then we can get the upper surface of the beak when we want to fit it to a parametric form automatically, and do this for the 150 meshes that we have across 50 species. So this is.
Actually, just on that question, why not use the homotopic thinning to get a medial axis in the first place? Like, is there an advantage to doing the iterative?
Good thing you're here. Yeah.
Homotopic thinning. No, I mean it's because arbitrary choice. I mean, it's just an arbitrary choice. Because in the end, they're the same by construction. Yeah.
Yeah, and we've done that for the antlers we did the shrinking, right? All right, so we have this. This is for Darn's finches, honey creepers, and their respective relatives. Okay, so now we could look at this, and we can sort of confirm from these 3D scans that this mid-sagittal section, you can really fit it to a parabola. So they're very well fit with parabolas. And then we can then look at the transverse cross sections. This one is like the basal cross section, and they're also very well fit with parabolas. And a more natural choice would have been a circle, but a circle doesn't work, and a quadratic fit doesn't work. So they're really parabolas. So when we try other fits that have the same number of parameters and the same symmetry, they didn't work. Okay, and then the other thing that we noticed that's interesting is that if I take this parabola versus that parabola, the curvature is increasing the closer you get to the tip, which makes sense, but it's also increasing roughly linearly. And we can then take that and use everything we learned to get this functional form for the beak. So we can look at the sagittal part, that's this parabola. Then we can look at, that gives us this curvature constant, the sagittal curvature. And then the curvature of the transverse sections, we fit it to a linear function. And so that gives us a tip curvature and a sharpening rate. And then this is a low dimensional morphospace. It captures the full shape of the beak. The beaks are not arbitrary shapes, so we were able to just find this functional form and plot them in this low dimensional morphospace. But one thing is that this still depends on scale. The units are millimeters here. So we want to sort of take that out of the equation. So okay, so when we want to do that, when I want to measure the depth and the width of the beak, and I look at how they did it in the field, they align first the beaks with the entire skull, the long axis of the skull. And when you do that, you get a certain one measurement of the width, depth, and length. And there's another way that we can do it where we just align with directly the direction of the beak. That gives us another set of measurement. And when we do that, so when we do that, we find that this way of doing it generates the variation that we see in the field. And this is actually, this dimension is the evolutionary significant thing that they measured. So when they measured that one population of birds is more adaptive, is more surviving like a drought because there's only big seeds, it's usually because it has a big depth of beak. And when you look at those birds, they actually have, is there a question? Okay, so when we look at those beaks, they actually are very angled relative to the skull. So just this tells us something about development and how you can generate this variation. You just have to change the angle of the beak relative to the skull. Okay, so once we have this angle, the dimensions of the beak, we can then get a dimensionless form of these parameters and then plot everything in those dimensions. And what we find, this was a surprise that everything went on a plane. And it turns out that the fact that they're all on a plane confirms that our fit works, that they're actually parabolas. If the fact that they're on a plane means that this section is a parabola. And then even more interesting, when we look at this two remaining parameters, the sharpening rate, how sharp you're getting towards the tip, and the sagittal curvature, you get that all the orange finches, their relatives, and the relatives of the honey creepers, they're all fall on one side of the line, and only the honey creepers who have high curvature are violating this sort of constraint. And we'll see that when we study development is this is actually because this is a developmental constraint. When you look at the developmental program of the beaks, we'll find that you can't generate anything with like Darwin's finches, the way they grow out here. And the way that Hawaiian honey creepers get over that is through curvature. All right, so to get closer to development, we're going to look again at these sections that we generated. We have sections and the center line, and we could look at the depth and width, and we can also look at the curvature of the center line. And we notice here another interesting thing. If you look at all the honey creepers, I'm looking at the curvature of this center line. So if you fit a curvature circle to that green line, green curve, you get the inverse radius of that. That's what we're plotting here in inverse millimeters. And what we find is that the honey creepers are actually, even this highly curved one, they're all around the same amount. So to our eyes, when you say something is really curved, you're actually not measuring curvature. You're doing a scale invariant curvature. You're looking at how much the angle is changing. And that's actually curvature times length. So you're generating this. So if this bird stopped growing faster, you would not call it a very curved beak, it would just be bulky. So that's another sort of signature of the way that they develop. If you want to get more curvature, just keep your developmental program going longer. Okay, another thing we can look at is how these cross sections are changing. That's going to give us another hint. So here I'm just plotting, I'm looking at this section and then something in the middle and something at the end. We see that they're getting more circular. And we can quantify that by looking at 1 minus the 4 pi area over perimeter squared. So when this is a circle, you get 0. When it's not a circle, you get something bigger than 0. And the similar thing we can do with convexity. So we see that over time, all the beaks are getting more and more convex. So we can quantify that.
So I lost track of where is time in this picture. So that's one beak, right?
That's one beak.
Yeah. So, and you are moving along the center curve and you got these 3 curves.
Sorry, I'm moving along. Yeah, so this is like
these 3 cross sections.
Yeah.
But 3 cross sections of a single beak.
Of a single beak at different places in the center line.
Yes. But you said something about time, like the growth or evolution. This is all about single beak.
This is single beak, and when I talk about time and single beak, I'm talking about development. So, and I'll show
biological.
Yeah,
not
yeah,
evolution.
not evolutionary time here. So these are different. These are. I'll show that in the next slide. These are different sections. Each one is generated at a different time in development. So this is generated first.
Not biologists. So where is the growth axis time here? We
Yeah,
started first and we started later.
Yeah. So
I see.
That's part of what we're doing is we are starting with a data set of adult or dead birds, right? So we're looking at scans of beaks, and from that we want to constrain something about development. So we're gonna look at actually how the birds develop, how over time their beaks change, and then map that onto the final shape. So we're saying that this part, the part at the base of the beak, was laid down first. Then this one was laid after that. So even though this is just a single beak, different parts developed at different times. Any other questions? Right. So going to development, this is that exact same picture. So these are different sections at different developmental times. So when you look at the beak, you see that there's a growth zone at the tip of every beak, and that growth zone has groups of cells that are proliferating. So those are the cells that are actually dividing. They're at the tip of the beak. And if you look at a different time, the shape and the morphology of the region that's the growth zone is changing over time. So, and that's happening because some cells are moving on and they divide and they stay in the growth zone. Some cells divide and they exit the growth zone. And this is like that same picture. Does that answer the question? Yeah. So the way that we're going to connect it is we're going to look at this cross section and say that that's the cross section that just exited the growth zone. That's kind of frozen. It's going to be part of the shape of the adult beak. And beyond this stage, what's been observed is that the shape of the beak doesn't change, it only scales. So we're saying this shape of these cross sections is being laid down at development at this stage. And so schematically, it would look like this. So we have, you're growing this, you have to control the direction that you're growing relative to your skull. Then you have your initial region, and that's evolving over time. And you have the growth zone is here. And when we map that on, this is an actual scan that we have. And then when we map that on into stuff. So this is how the evolution would look like of the beak over time.
You mean
Development, yeah. Evolution over time or development. Yeah. Okay, so we want to see how this is generated. And one thing we notice when we look at this video is that, so from the scans of the beaks, we see that this region or that region is moving at a different rate compared to this highly curved region. So the reason why you're becoming more and more convex is that this part is being chipped at faster. So those cells are exiting the growth zone faster than cells in the center or cells over here. So, and that's, to explain that, we want to see how cells just know where they are. And we propose this morphogen model, which you have reaction and degradation. So that gives you a length scale for the range of this morphogen. And the likely candidate of this is calmodulin pathway, because that's the protein that, or the signaling pathway that's linked to longer beaks. So when they overexpress it in chicks, the chicks grow longer beaks at the right stage. And I think that's because it's controlling whether cells stay in the growth zone or exit. Okay, and then we're assuming there's this layer that's kind of the leading edge of the growth zone. It's producing this morphogen. All cells are producing at the same rate, but the cells are on this side, or on the highly curved regions, they have less neighbors, so they will experience less gradient or less concentration of that morphogen. So they're exiting the growth zone at a higher rate because they have lower concentration of that. Okay, and so the model basically assumes that you have a background rate of cells just exiting the growth zone, but that's enhanced by the concentration. And lambda here is just the range of this morphogen. And then near the boundary, we can calculate that this concentration will be a linear function in curvature. And this is what it would look like if we run the model. So this is the leading edge of the growth zone, and each cell is deciding whether to divide or not based on a threshold. And you see, you start with this basal shape, and then it's becoming more and more circular over time and more convex. And that's the sort of the fitting. Any questions about that? So then so this is kind of just simulating it at the cell scale. We can also just zoom out and look at the scale of the tissue. We just have the fact that the rate at which this region is moving, the velocity sort of exiting the growth zone, is a linear function in curvature. And that's called, that behavior is called the mean curvature flow. So the regions over here, the velocity is smaller. Over there, you have higher curvature, so you're kind of moving faster. And from our actual beaks, where the t and t plus dt are actually just measurements along the length of the center line. And we see that the velocity is actually a linear function in curvature. So, and we can also simulate that at the tissue scale and also get a good fit and do it across all the samples that we have. So this is all the species in Hawaiian honeycreepers.
Does the reaction diffusion equation idea also imply that as the peaks get thicker over time, as they just get larger in the orthogonal plane, that they would also become more convex or more circular? Like this idea of moving to the next place forward.
So we're assuming that they're not getting bigger.
I see.
But I think you're right. Like because it's not reversible, like it's not time reversal variant. So if you go in the other direction,
I think Sean's question was no protrusion, just a single curve, and asking how to do that, right?
Yeah. So if moving forward in time to the next that way, that would also be here
At the same location.
at the same location in time if there's a growth in the plane, and
yeah,
then you'd expect over time that it would do the same thing. I guess.
yeah.
If it behaved.
Yeah.
Yeah, that's the question. Yeah.
Answer the question?
Yeah.
Yeah, so so you're asking if it's growing, it also becomes more circular.
Yeah.
Yeah.
Or if that would be an implication, or why that wouldn't be an implication from.
Yeah.
Okay.
So what we're I guess I assume that in the what follows in the developmental constraint that you're shrinking. But so that you're not expanding, that the growth zone is not growing. But yeah, you're right. All right, so this is another example of a fit. And so just to put it together, we have this generative morphospace of beaks that's inspired by their development. We need to consider the basal cross section. That's your initial condition. And if you look across all the species, all our samples, these are all like what the cross sections look like, scaled by depth and width, and they mostly similar. So they have a similar kind of shape. And we quantify, like, so if you put the depth, width, and this curvature, the length of the beak, plus this initial condition, that gives you all the beaks that we have. With what's varying, dimensionless parameter quantifying this mean curvature flow. So there's one, that's what I was plotting here. So this is the only parameter that's quantifying the different beaks, apart from just length, width, and depth. And the one thing that I haven't considered that's different from the mean curvature flow is how are you getting the centerline curvature? So that's an independent level that's introduced maybe either in Hawaiian honeycreepers or in other species that's causing proliferation to happen more at the top than at the bottom so that you get these curved beaks. Okay, and last, what I mentioned earlier, that we have this constraint that comes from matching this mean curvature flow to the parabolic fit that we found earlier. When I put those two together, we get that if I assumed the cut that I made for Darwin's finches, we get this condition. And then for honey, when we tested it, we saw that Hawaiian honey creepers don't satisfy that. And that's basically when we updated our analysis to account for the entire curvature. And if you now derive a constraint that accounts for that, this is a ramp function. So it's 0 when this is 0, when this is less than 0. And this parameter is just telling you, like, if you look at the beak, how much is it curving at the edges of it. So like it's higher for Hawaiian honey creepers. And so it's only positive for Hawaiian honey creepers. It's negative for other species. And when we account for that, we get a better fit of the constraint. So I could say that all these samples are here, but we could easily, from our parametric morphometric, say, what would a beak here look like? So if you assume flat, it would look like this, flat edges. If you assume curved edges, it would look something like this. But those are beaks that we would say from development never occur, from considerations of development. Okay, so I'll just close here. We in either way that we did it, we have two-dimensionless curvature parameters plus the depth, width, and length of the beak that sort of capture all the variation. And so the dimensionless parameter here was just the curvature of the center line and the parameter that characterizes how these cross sections are evolving. And we have this developmental constraint that also follows from connecting the two different morphometric methods. And what's still missing is more direct confirmation of more experimental verification of the model, looking at transverse sections. The data that we have was sagittal sections, looking at how the growth zone is evolving, but we would want to be able to look at transverse sections and look at the directions where cells are dividing so we can constrain the model. Sorry, these are loading slower. And even more dramatic bird families, this is Madagascar vangas, would be great to test the morphometric methods on those. And one thing that me and Gary were thinking about is we've done some of this, but I didn't talk about it here. Like looking at the entire skull, looking at the skull biomechanics, correlating things with diet. And an important breakthrough might be to be able to, because there is one manual method step, which is cutting the beak. If we automate that, then we could do this for thousands of skulls at the same time. And thank you for listening, and it was a great pleasure.